Okay, this is the second lecture in Unit 6, the cell cycle. On the last lecture, we described the M phase of the cell cycle, and now we are going to analyze what happens in the S phase of the cell cycle. So we're looking at our second learning objective here. Okay, so the S phase of the cycle, or our second learning objective, deals with DNA, and DNA stands for deoxyribonucleic acid. The word is going to use DNA for short. And we've studied this a little bit earlier in the year. We learned monomers and polymers of different organic compounds, and DNA is one of the nucleic acids. That's why it's called nucleic acid here. And the monomers of the nucleic acids are nucleotides. And nucleotides are composed of three parts. They have a 5-carbon sugar, and so remember when we were drawing sugars earlier in the year, we used different shapes. So this would be a pentagon here, right? So this would be the 5-carbon sugar. And then it also has a phosphate group attached to it. So this is representing the phosphate group. And then in the middle there will be this nitrogenous base. So a polymer would be all these little monomers like this linked together, and it actually will go sugar, phosphate, sugar, phosphate, sugar, phosphate in this long chain with these nitrogenous bases sort of sticking out from that chain. And that's what makes the DNA a polymer. So the DNA chains, the way that these nucleotides are arranged, is in the form of a double helix. So this right here, if we follow this strand, this is a helix. It twists back and forth. But DNA is composed of 2 helices like this. So here's the other strand running in a helix. So we call that a double helix. And what holds these two together are those bases that stick out. So here we go with our sugar and phosphate, and sugar and phosphate, and sugar and phosphate. And that's like this ribbon on my DNA here. Then the bases are these yellow blobs, and they're sticking out in the center. So this strand has little nitrogenous bases sticking out in the center, and this strand also, and those nitrogenous bases bond together, and that's what holds these 2 DNA strands together. So we have these hydrogen bonds, which are weak associations, easily broken, which we're going to see why that's important later, that are holding these 2 strands together. So these bases are kind of what's important because they're what holds the code for all the proteins that your body makes. So the sequence of these bases are going to control what proteins your body ultimately makes. And we will learn about that in another unit on protein synthesis. But right now we're going to learn how we replicate this molecule. So the bases are A, T, C, and G, or adenine, thymine, guanine, and cytosine. And the way these bases are held together is very specific. A always binds to T, and G always binds to C. So if you have one strand and you know this is a guanine, you know that this will be a cytosine. And if one is a thymine, like here, we know that this one's going to be an adenine. And so by looking at one strand, you can predict what the other strand would look like. And that's actually how it gets copied. So one way you can remember this is that A's are straight lines, and so are T's, and so they go together, and C's and G's are curvy lines, so those go together. So when the DNA replicates, what it does is it separates these 2 strands. So those hydrogen bonds that are holding those bases together in the middle break, and the DNA unwinds, and so we have a single strand here and like a single strand there. And once they're open, little nucleotides, those monomers, are floating around inside the nucleus, and they will be attracted to the open base. So if this G doesn't have a C attached to it, a C monomer, just the C and the sugar and the phosphate, right, the monomer, that's the C will be attracted here and will bind. And so you make this strand. So it slowly builds this strand. So this T will eventually attract, this A will attract to T, this G will attract to C, and so on. And so that you build the 2 strands. And when the entire thing is done, you now have 2 strands that are identical to the original strand that you started with. So you have no changes in your DNA, but you have 2 identical copies. And then those 2 identical copies will coil up but when they coil up, they will stay attached to each other at the centromere. And then the cell will be ready to divide. Okay, so in this process of matching up these A's and these T's and these C's and these G's, mistakes happen. But your body has ways of going back over it and making sure there are no errors. In the end, you end up with about 1 error for every billion base pairs, which is pretty incredible. Mutations, however, do happen. Changes in the DNA is a mutation, and that happens, and especially if the organism is in an environment where there are different chemicals or different things that we call mutagens, things that change the DNA. Even UV radiation from the sun can do this. Some mutations are good. Rarely they're good, but sometimes they're good. Most of the time the mutations are bad, and sometimes they're indifferent. But if we didn't have any good mutations, we wouldn't have any change in organisms over time. So all of the differences that we see in DNA in different organisms was due to some point, some mutation that was good for that organism. Oftentimes, though, they're bad. Sometimes they're indifferent. They're in an area that doesn't really make a difference. Okay, the other thing that's really important in the cell cycle, and we're going to kind of wrap up this unit, is that the cell cycle itself is under very close regulation. So in other words, here we have our cell cycle. These little arrows represent areas where the cell cycle has to pass a checkpoint before it can continue. So the S phase here, right in the middle of this DNA replication, there's a point where it needs to be checked before it can continue. Right before it goes through mitosis, it's got to be checked. In the middle of mitosis, when all the chromosomes line up, there's a checkpoint. And if we didn't have these checkpoints, your cells could divide and divide and divide and divide. And if that happens, you have cancer. So uncontrolled cell growth is cancer. A tumor is a bunch of cells that just divide and divide and divide. And oftentimes they don't specialize. They don't end up being the kind of cells that they're supposed to be. Sometimes they can become very bizarre cells, but they're not going to be functional in doing what the organism needs. And they may end up building a huge mass that puts pressure on that organ, may put pressure on a blood vessel or on a nerve, and ultimately that can lead to death. It can also metastasize, or those cancer cells can break free and travel in the blood and go somewhere else in the body and start to divide out of control. So controlling the cell cycle is really important so that you don't have cancer. And when those checkpoints that I was talking about don't get adhered to, something happens and something passes a checkpoint when it's not supposed to, that's what leads to cancer. So we just did the 2 learning objectives. In the last lecture, we did the M phase of the cell cycle, or the cell division, mitosis, and then following that with cytokinesis. And then we've just talked about the S phase and how DNA is replicated. And then just remember that those 2 G phases are growth and the cell just doing its cellular job, whatever that happens to be for that cell. And that is Unit